Hello, everyone, and welcome to BrainMap. As you all know, BrainMap is a seminar co-sponsored by the P41-funded Center for Mesoscale Imaging, housed in the Martinos Center. So it's my great pleasure today to introduce our speaker, Dr. Jon-Fredrik Nielsen. Dr. Jon-Fredrik Nielsen is a research associate professor in the fMRI laboratory at the University of Michigan, specializing in MRI sequence design and implementation. He created and manages the PulseSeq interpreter for GE scanners and is the co-PI of the harmonized MRI project that seeks to implement and disseminate vendor-agnostic, functional, and quantitative neuroimaging protocols for more reproducible MRI research. We are most honored to have you here, John, today. And without any further ado, just going to hand it over to John. Everyone on Zoom and here knows the drill. John has agreed, if you want to ask questions, just go ahead during the talk. Please, people on Zoom, you can raise your hand or just put it in the Q&A box, and people here, I guess, will just shout out the questions. So, John, thank you so much for being here.
Thank you. Thank you so much, Shabazz, and thanks for having me. Thanks to Birkin for hosting, and Shohei and Sean and everyone else who helped me out. We got some data yesterday, and that I got to tour Boston. Some of the MRI scanners. Seems like there's an infinite number of them. But it's been really, really great. Thanks for the collaboration. So I'll talk about our progress toward harmonized environment for multi-site fMRI. I actually won't show any fMRI activation mask, believe it or not, but I'm going to describe the tool that we've developed. And my goal here is to hopefully make you interested and willing to try it out and test it for us. So I'll try to give sort of the broader picture generally, talking about vendor-agnostic sequences and what we call the harmonized MRI project, which I'm working with Birkin and Yogesh on, and Maxime as well, Maxime Taxit, and others. And I'll talk more about that. So that's kind of the umbrella project. And then I'll talk specifically about SMS EPI, functional MRI, talk about the acquisition sequence and the image reconstruction. And that's basically the tool that we provide. And it's based on the PulseSeq vendor-agnostic sequence format. So that's the essence of this. And if time allows, I'll talk more about the PulseSeq and the general idea and how it works and the PulseSeq user community. So the general framework is applicable to not just functional MRI, but other applications as well. And our folks here have applied it to diffusion and quantitative imaging. And I'm going to talk specifically about where my intended target audience for this talk is functional MRI. So if you're an fMRI researcher interested in pooling data from multiple sites, or a sequence developer who just wants to try out a sequence, either that sequence idea that you have, or perhaps you want to try out that someone else has come up with, this is a good framework and tool for that. So that's kind of the overview. And we're looking for people to test this out. So kind of in a beta testing mode. So taking a really big view of the functional MRI workflow. So there's some kind of process, biological process that happens that we'd like to image. And then we, quote unquote, acquire the MRI images. And then we analyze those images. And so that's kind of the very high level picture. One of the primary motivations for this work is to try to minimize the variance in the data, in the measurements they ultimately obtain. And so I've tried to break this up kind of crudely into the various components. So there's variance just from biology, which we can't do anything about. And often that's what we want to measure. So specifically, we'd like to measure some variability between subjects. And that's often the target of our functional MRI study. There's generally also variability across sessions. So suppose you put me in different sessions a few hours apart, there will be variability there as well. So that's important to keep in mind. And we can't do anything about that. We can try to, if the goal is to minimize this session variance, we might need to characterize that. But ultimately, we hope to say something about the functional activation in a particular set. So, and then we must acquire the images, and there's going to be some kind of variance between sites, so site-to-site variance or inter-site variance. that that is a nuisance. So we'd like to reduce that as much as possible. I'll say a bit more about that. And then on the analysis side, depending on what analysis pipeline or analysis tools that you use, that might introduce some variance, let's say across, if different groups, as we know, analyze the same data, they can come up with, different conclusions. So there's been a lot of work, of course, by this institution and others on harmonizing, the analysis side, with tools like FreeSurfer and FSL and so forth. And so I think the open source community has really rallied and provided amazing tools on that end. So I think, in a sense, managing that and minimizing that is something that the field has addressed and come up with really, I think, mature solutions to. Of course, it's still an ongoing process. Acquiring this variability and the acquiring the images is really what I want to focus on in this talk. And so, leaving aside any other variability, we'll have to accept that it exists, and if needed, design our study to measure those. But we hope that we have a method here for minimizing the site variance or inter-site variance to the greatest extent possible. And so that's kind of where our work fits in, right in the middle here. And so if we look at that in a little more detail, so what does acquiring MRI images mean? So it means to implement the whole sequence that runs on the scanner, calibration scans, core calibration scans. So you have a choice there as to what calibration scan to use. And of course, you have to acquire the data and then reconstruct the images, and potentially there's some pre-processing like Gibbs ringing, reduction or something. So that's kind of the whole process. So, and once you have those images, you can analyze them. Each step here potentially impacts image quality. And at the moment, we, for the most part, leave this to each vendor. So the vendors, just to acknowledge that, of course, the vendors have provided incredible hardware. And also on each vendor, the tools are really, push button and easy to use. They produce images that are useful. The vendors probably don't have a large incentive to harmonize across different vendors. And so that's kind of where we come in. So the current practice is to kind of treat this as a black box. It can be, difficult or impossible to get all the details needed here to fully understand, how each vendor implements, the whole sequence, all the low-level details, and these other, certainly the image reconstruction. And so a lot of that is kind of hidden for us researchers. And so what we're doing, we're taking a kind of a vendor-agnostic and open source approach. Our goal is to make all these components as open and harmonized as possible. And so that's where this work fits in. So it's nothing more or less than essentially opening up that acquisition and image reconstruction process and making sure that how the data is acquired and reconstructed. And so we're making this tool available, and hopefully the community will contribute back, to optimize this further. So that's kind of the big picture. What are some high-level approaches to harmonizing the protocol? So I think there are, you could say, generally two ways. One is to limit the study to MRI scanners from just one vendor, which is kind of a brute force model. But that is certainly a good approach, and there are studies that do this. Even University of Michigan, we often bring patients to the same scanner for this reason, just to avoid the practical difficulties of dealing with different sites and eliminate that potential variance. So that certainly works. Of course, you have to maintain the protocol across different software versions, and different software versions might also have slightly different implementations of nominally the same sequence. And also sort of, at least theoretically, you don't generally know then if the findings that you have obtained only apply to that specific model, or if it might break on some other vendors' skin. The kind of the deeper level approach that probably has a better chance of working overall is to include multiple vendors, but then to implement custom pulse sequences from scratch, more or less from scratch, or at least to some extent on each vendor platform. Or just dealing with the product sequences and then trying to harmonize. This would require expertise, both programming expertise, and that usually involves vendor involvement. And of course, now you have a lot bigger maintenance task because you have to deal with more vendors. And even after you do that, this may not ensure that sequences are identical at the end of the day. So depending on what level of harmonization that you're willing to invest time in, either just optimizing the high-level parameters like the matrix size and so forth, or if you're willing to actually go in and ensure that sequence details are the same, things like what fat suppression technique to use and slice profile and other things. And so what I want to acknowledge here is that we don't actually know the extent to which these efforts are really sufficient to reduce the inner site variance enough, quote unquote, enough for a particular application or context. And so my hope here is to basically just say that we're providing a tool, the technology is now mature. And now the next step then is to, evaluate and see, try to determine what are the factors that contribute to the variance across sites. So at least now we have a way to do that and control that. So I just want to kind of make that point clear. All right, so let's step back. So if you want to do pulse sequence programming today on multiple vendors, this is, I'm sure you're all familiar with this, but this is basically what you need to do. So suppose you have some desired pulse sequence, spiral, whatever, EPI, whatever you want to implement, or a standard method that you want to implement in a way that, so that you know exactly what all the details are. So if you want to implement that on two vendors, A and B, you then need to implement that separately, independently on each vendor, using that vendor's pulse programming environment. So that produces an executable that then works on any particular vendor's platform. So in the end, you have some concept about the sequence, and then you implement that independently. And this is rarely done by the same person. So this usually involves multiple people and possibly vendors. And even then, at the end of the day, you're not really guaranteed that the whole sequence is identical. Let's see. Yeah, I think I somehow moved. Oh, there it is. Okay. I think I jumbled those last two slides. But what we'd like to do then is for one person to implement that sequence in the vendor-agnostic way. It could be a text file or some kind of vendor-agnostic format, and then have interpreters on the multiple different scanners to quote unquote just interpret that and play that up, convert that to hardware instructions in a way that guarantees that the full sequences are identical. So that's where we want to go, and that's basically what this tool allows. So nothing more, nothing less. Let me just check if there was any. Oh, I just wanted to say that I want to focus on the data acquisition piece first. And so, just a very brief and very busy overview here of other or various vendor-agnostic pulse sequence programming tools that have been proposed over the years. And this goes back really more than 20 years ago. And so there are many really amazing, actually, tools that were developed, and they have stuck to greater or lesser extent over the years, depending on various factors. So there was, for example, a Java-based complete tool developed internally by GE that was essentially also portable, but it depended on the Java API, which had certain drawbacks. There's Odin that was demonstrated on multiple vendors. Of course, there's the commercial offering that's very much an option and alive today, which is Vista. There's Sequence Tree, another platform that I believe primarily has been implemented at Siemens. And there are other, and that brings us to today. GammaStar is a is a setup where you can also provide vendor-agnostic sequences in an interactive way, and that distinguishes it from the pulse-seq approach on the right here. Here, the idea is to basically implement the sequence in a file and implement and define all the low-level details in that file. So once that file is designed, that's fixed, and you just play it out on the scanner. So you don't have that interactivity on the console. So that's kind of the main difference between these two. But these are both being pursued today. So we focus here on the pulse-seq platform, just mainly because of the ease of defining the sequences in a kind of a human readable, neutral way. And so I'll describe that. So the basic idea, high-level overview, again, is to design or create a sequence file that defines the pulse sequence in all details. So it defines all the low-level events. And that can be created using various tools. So you could, in principle, write it by, well, it would be very hard to do, but you could write it by hand if you want. But you can use the pulse-seq MATLAB toolbox or the Python implementation of that toolbox to create pulse-seq files. There are also other ways to do it: GPI Lab, GEMRIS, and then all those other tools. Even GammaStar, actually, you can export pulse-seq files. So there are various ways to implement that file. Once you have it, then the idea is to implement it, to send that to an interpreter on different vendors and quote unquote just play it out. So that's the idea here. So you completely separate the heart rate independent part from the heart rate dependent part. And this has been implemented on other systems like low field scanners and so forth. So this is meant to be very kind of generic. And if I have time, I'll talk a bit more about the specific format later. So that brings us to what we have named the Harmonized MRI project. So this is kind of a broad overview. I realize for the audience, I'm blocking the URL there, but hopefully you guys on Zoom can see that. So the idea is to use pulse-seq to design the sequence, and that will create the sequence file for whatever application you have in mind. And then we acquire data. So at the moment, it's fully supported on Siemens and GE. A Philips interpreter is in the works, and I'll talk a bit about that in a minute. And then you take the raw data file in whatever format that you choose. And we provide tools also for converting the vendor sequences to, for now, we have our own little custom H5 format, but that's something we can easily modify. And then image reconstruction. So that's the basic kind of workflow that works for any pulse sequence in any application. So the team consists of several sites. So it's Maxim Zaitsev's group at Freiburg, who of course developed the pulse-seq standard and are managing the Siemens interpreter. And there's our group. We developed the fMRI protocol and the GE interpreter. And there's folks here in Boston and Case Western. And I should probably have more logos here. There are so many sites here, and I'm still learning what they are. Here in Boston, they work on applications like diffusion quantitative neuroimaging, and also the Philips interpreter. So, and the funding at the moment comes from these two grants. This is also very much a community effort. So this is just kind of the core team to really get this started. We really want this to be used. And so having you try any of this and giving us feedback and then using it for your needs is really what's going to drive this forward. So I'll give a specific example, SMS EPI example, and I will try to give enough detail for you to actually go and try this out. Well, first to perhaps make you want to try it out, and then enough detail to sort of explain that this is something that you can do, and we want you to contact us, and we can give you example data and other things and to make it work for you. So that's, I have a website here with a specific example that I'll go through. So that's the goal. So here's an example image that you would obtain in the end. So on the sequence design side, you can either, actually I don't have a download link yet, but you can just contact us and we'll send you a full seq file, such as the one I just tried yesterday here on your scanners here. But if you want to create your own, you can do that, and we provide a script for that. At the moment, that script and also the recon script is in a private GitHub repository, but we're happy to share it. So just send me your GitHub username, and that helps us keep track of usage, which is useful for us for reporting purposes. And it also ensures that we want to make sure that we don't, trample on any IP here. We're well aware that we're not the originators of this sequence of reconstruction. So for now, if you want to use this just to try out, that's totally fine. If you want to use it for functional MRI studies, we ask for now at least that you also have the vendor SMS API protocol on that scan, so that the vendors are supportive of both Pulse project and specifically the fMRI U24 that's funding this. So they're fully on board with this, but we just want to make sure that we don't take away any business from the IP holders. So this is the sequence file. This is just an example. So you can take that pulse, the seq file. This is an SMS EPI sequence with a fast saturation pulse, SMS excitation, and EPI. So there's nothing special. This is essentially implemented kind of in a standard way. This is actually simulated using GE's Pulse Studio, which shows you the exact timing of the sequence. And so it just shows you that it's a sequence that you can use the vendor's simulator tools to implement. You can do the same thing in IDEA and get a plot, and then you can inspect the sequence in detail to ensure that you get what you want. And this also is a first way to ensure that we really get accurate translation of the seq file to the vendor hardware instructions. So once you have that seq file, you go to your scanner and run it. And I'll talk a little bit about what you need to do in order to do that. So if you're a Siemens user, you have to install the interpreter. I'll give some details on where you can get that on the next slide. You place the seq file on the scanner, you prescribe it, and then run it. And that's actually it. So it's a very direct kind of translation, very direct interpreter. The Siemens interpreter requires the C2P mechanism. It's a universal platform-independent source code. And you compile it once for each of your different scanners for a given software version. And then you're done. You can play multiple seq files. So the interpreter is sequence agnostic. This comes with a PDF manual. And as I mentioned, once you have a seq file, you can simulate that in IDEA using the vendor's tools. And you can contact this email here if you want to get that interpreted. On the GE side, again, give details on the next slide how to get that. There's a file conversion step at the moment that's implemented in MATLAB that converts the seq file to another file, actually a tar file. It's a collection of files that are then put on the scanner. But it works, apart from that, it works in the same way. To obtain the GE interpreter, you can contact me. This is also a universal platform-independent source code, at least for version 28 and up. And we can send you code or the binaries. We're happy to compile it for you. As with any research sequence with GE, your institution must have signed the research community sharing license, which chances are it probably has. So that's a general license for that allows research users to share basically epic source code. Comes with a user guide, and then you can simulate it in Pulse Studio or whatever the tools as I just showed. So it works pretty much the same way as the Siemens tool. As I mentioned, there's some really exciting work on adding a Philips interpreter for PulseSeq that come pretty far. There are actually 2 groups that are working on this independently, and I listed them here, and they both gave presentations at the ISM virtual meeting that we held in November on vendor-agnostic pulse programming. And those recordings are available here, and also the slides from all the talks and all the program overview is available on this PulseSeq GitHub page. So I urge you to have a look at that, and there are lots of talks on introduction to PulseSeq, and also on applications from the user community. So we're pretty excited to see PulseSeq being put to use by the community. I'll say a little bit about just the first interpreter, just to give you an idea about what's involved and how it kind of contrasts with the Siemens and GE interpreters. So this is probably a bit more, maybe too much detail for those not on Philips, but again, the idea is to create your seq file, and once you have it, there's also a sequence conversion step where you take that seq file and create another file or set of files that are then loaded onto the scanner. There's to get around some of the hardware constraints there are always going to be different constraints on different vendors. There's some internal logic that combines blocks if possible. So for example, in an EPI sequence, it would actually not generally be possible to put each echo right one next to each other. But if you combine them in an intelligent way, you can do that. So this conversion takes care, and the interpreter takes care of that mechanism for you. So here's some initial results. This is create an echo acquisition with Philips and with PulseSeq on Siemens. It's the basic sequence, and also using diffusion-weighted EPI. So this works, and I'm personally not evaluating this yet, but other folks in the group are. So if you're interested, we'll actually contact either of the authors of those two interpreters to try it out. Yeah, so specifically, if you want to try out Shake's interpreter, here's the contact information. Okay, so that brings us to the image reconstruction step. We provide two ways right now of reconstructing the SMS EPI data. The one that we're probably using most at the moment is a slice grappa reconstruction. I want to thank Birkin and Kawin for a lot of help and example code. The implementation is basically, it's our own re-implementation of the slice graph that we're basically. This is, the details here are not useful, but the basic idea is that the slice graph reconstruction is available in this MATLAB namespace. Basically taking the acquired data and the 2D slice calibration slices, and then reconstructing the data. And so this is kind of an initial version. There are some parameters that one can choose. So this is not set in the stone, and we expect continued improvements, things like regularization factor and other things, full compression and other things that we can look into. But this is where we hope that the community can give us some feedback as well. An alternative that we have also implemented is a 3D sense, and that accounts for B0 inhomogeneity. That's written in Julia, so one nice thing about that is it's open source. So this is just an example where this is not from a steady state fMRI sequence. It's just an example to show that it works. So there's a 2D reference slice on the left and the multi-band factor 6 image on the right reconstructed with an essence, with 3D sense. So two versions of it, and you're welcome to to have a look at either of those. Just as an aside, we're also working on the 3D stack of spirals, so we have reconstruction code for that as well in Python. So the details of this example are provided here. So maybe I can briefly show. But this example is intended to give you basically all the details that you need in order to understand the workflow and also to try it out yourself, to implement your own version of it. So it includes all the things that you need, converting the raw data to something that you can read in a vendor-agnostic way, and EPI RAM sampling, reconstruction, and ghost correction. There are a couple of additional sequences that you need. But you can customize this to fit your own workflow. And this is just an example of whole brain slice graph reconstruction. Maybe I can quickly see. Yeah, so this is the example folder. So again, this is the, you can see that I'm at the harmonized fMRI API. Perhaps it's a complete example. Oh, it's a little slow, it seems. This is the main landing page. And then the example is where you want to start first. So slow. You can look at main.m, which just gives you an overview. So I've just bundled, these are just scripts that you can look at, that you can see the workflow. If you're on Linux, you can actually run this and get all the code from GitHub directly, or you can do it manually if you're on other platforms. And then you set the parameters. We have a script for reading the raw data for G and Siemens at the moment, and then converting that to a file. So after this point, everything is vendor agnostic, vendor independent. This goes calibration and getting the 2D multi-slice data, and then reconstructing the time series. So if you look into the details of the script, you can see the steps that are needed. And we can provide you with example data that when you try it, you'll end up with an image like this. So that's kind of the SMS CPI workflow. And yeah, as I mentioned, we'd love for you to try it out and give us some feedback. A little bit more about PulseSeq. PulseSeq is it's a general purpose language, not just for SMS CPI, that describes MR pulse sequences. It's also the software to generate the pulse sequence, specifically all the tools that I mentioned, MATLAB or Python. And it's also kind of an ecosystem and really a kind of a community to generate sequences and the software to play them on. Yeah, so the idea philosophy is minimize effort to implement, easy prototyping, and make things like arbitrary gradients, arbitrary RF easily accessible to researchers. And I think an important part that I really appreciated in the MATLAB PulseSeq toolbox is that it has error checks, so it's very easy to make simple mistakes regarding the timing and things when you put things together. And it's the toolbox catches a lot of those, so that's an important aspect. And overall, to promote open source thinking, sharing, and exchange. And so the basic idea of PulseSeq, and I think is useful to kind of realize this basic block concept when you even when you use them, so that you know and understand what's possible and perhaps what's not. So it's a sequence, or the sequence is represented as a concatenation of non-overlapping blocks. And gradients, they don't have to start or end at 0. So here's an example of how you can define or split a sequence up into independent blocks. This is just one way to do it. You could do it differently. that those first two blocks there could be combined into one. So it's a little bit up to you. But that's the basic idea. So kind of thinking about sequences in terms of these blocks is kind of the starting point for how to put the sequences together in PulseSeq. And so here's an example of a MATLAB script that would generate a PulseSeq file. So this is essentially basically all you need. I think this is the entire code to generate a 2D gradient echo sequence. So the idea is to specify the system limits. There's a built-in function for creating an RF pulse excitation. You do some sequence timing calculations. Then you have a loop where you call this add block command, and I will just add another block to the sequence. So this block concept, it allows for very compact code for creating those sequences. So here's another example. Is just to show that the MATLAB toolbox gives you a tool for displaying these. And this is a very, very general framework. So you can play in principle anything. And I couldn't resist. So what I'm going to do is play some music for you that is created by the scanner. So this is again using the MR Music PulseSeq toolbox, creating a PulseSeq file. And then admittedly, I had to do a slight adjustment on the GE interpreter. That was just due to the way that I had implemented the interpreter, and it's not a limitation of the hardware in order to play this, to get this to work. But let's see if it'll play, and I hope you'll hear it. This is playing arbitrary waveforms on 3 gradients. You have three notes to play with, and then Maxime put this together and played it on his Siemens scanner, and then I couldn't resist and did it on our GE scanner, instrument machine as well. So this is a 1-minute scan, and since the hardware, from different vendors has very similar performance, and they're internally capable of more or less the same thing, this is very doable. But this would be, impractical to implement using standard techniques. So this demonstrates how flexible this framework is. There are, as I mentioned, some good PulseSeq resources out there if you want to learn more. We did the vendor-agnostic pulse sequence programming course, as I mentioned, 3 days, and they're all recorded. There have also been workshops before called MRI Together, both in 2022, I think, and 2023. And those talks are also recorded, so I would urge you to check that out. So I will summarize here. So I would say that the technology exists for truly harmonizing fMRI protocol down on the, basically the individual waveform sample level. This is useful for harmonizing fMRI protocols and other protocols, but also for rapidly prototyping new sequences. And there's a growing PulseSeq user community. I think you'll see a lot of presentations like ISMRM this year. And people are really starting to put this to good use for a range of applications. The key step for us on the functional MRI side is to use this tool to begin to evaluate cross-site reproducibility, and we're just starting to do that now, doing some traveling subject studies at University of Michigan. And so the open question here is what factors are important to control for, and what are the impact of this? So this is a little bit of an unknown, but I think that's the big next step and big challenge for this project. So we spent last year, I spent a lot of time updating the GE interpreter, basically rewrote that to make it as accurate as possible. And I think, and also working on the reconstruction, I think now we're at the point where we can really start to put this to use. So I want to acknowledge a lot of people at these institutions, and there are many also folks that are using this and that have contributed in various ways as well. But this is kind of the core harmonized MRI team. Yep, I'll leave you with that. And, thanks very much for your time.